Hello and welcome to Conversations on AFM. I'm Dr. Elizabeth Kugler, a science communicator and your host of today's conversation, Nanoscale Chemical Analysis with AFM-IR. Today we are focusing on AFM-IR, which is a technique that integrates atomic force microscopy with infrared spectroscopy to deliver nanoscale chemical analysis. This powerful approach drives breakthroughs in material sciences, life sciences, and beyond. And so to discuss this really exciting topic with us today is an incredibly esteemed guest, which we are really happy to speak with today. Dr. Craig Prater, who is the CTO and co-founder of the Photothermal Spectroscopy Corp. He has served as a visionary leader in active AFM development, and he is a co-inventor of many patents in both AFM and AFM-IR. He has performed photothermal spectroscopy research since 2007 and has led the development of optical photothermal infrared technology since 2017. In 2023, he was recognized with the Coblentz Society Williams-Wright Award for significant contributions to vibrational spectroscopy while working in industry. And he has co-authored more than 140 publications, including scientific papers, trade publications, and patents, which together have accumulated well over 10,000 citations. So it's my pleasure to welcome today Dr. Craig Prater.
Thank you so much. It's a pleasure to be with you.
Awesome. So we really dive straight into the topic, and we first want to learn a little bit more about what is AFM-IR, because we know that AFM is a really broad topic. And in previous episodes of this podcast, Conversations on AFM, we have explored different subtypes of AFM to study biological and electromechanical features. But today, we really want to explore a slightly different aspect of it, which is, as I mentioned, AFM-IR. And this is to provide nanoscale insights about chemical information. So, Dr. Prater, you have been an absolute pioneer with this, and you have really led the commercialization of AFM-IR. So it would be great to learn a little bit more about what is AFM-IR and how does it work?
Sure. So as you mentioned, AFM-IR is a combination of two well-established techniques, atomic force microscopy and infrared spectroscopy. And backing up a little bit, I want to talk about a little bit about the motivation of AFM-IR, and then I'll talk about how it works. So for an extended period during the development of and use of AFM, there was a very common challenge where AFM really excelled at achieving very high spatial resolution, making beautiful images of a wide variety of samples. But one of the most common questions is we would look at an AFM image of a sample and we would see some interesting structure. We would go, well, what is that? And the AFM was not very good at answering the question of what is that? And we proceeded to try over the years a wide variety of techniques to understand more of the what is that. We would figure out ways to probe the electrical properties, the mechanical properties, the friction, the adhesion, other topics that you may have discussed in previous podcasts. And all of these approaches gave us information about what the material, but it didn't give us any kind of unique identification of the chemical composition of the material we were looking at. And so what AFM-IR is a technique that solves that problem by using the chemical specificity of infrared spectroscopy and the high spatial resolution of AFM. And specifically how it works is there is typically a tunable infrared laser that is aimed at the region of the sample where the AFM tip is touching the sample. And if the tunable infrared laser is tuned to a frequency or a wavelength that corresponds to a infrared absorption band in the sample, then part of that laser light is absorbed as heat. And specifically what's going on is that if the vibration frequency of the light matches the vibration frequency of molecules in the sample, then part of that light goes into exciting the molecules, causes them to heat up. And every time there is a laser pulse that causes the sample to heat up, it correspondingly causes a minute thermal expansion in the sample. It basically causes a kick to the tip of the AFM that causes some motion in the tip of the AFM that can then be detected using traditional AFM techniques. And then if we sweep the wavelength of the tunable infrared laser while measuring the AFM cantilever motion, we can create an infrared absorption spectrum that basically shows increases and decreases in the amplitude of the AFM cantilever as a function of the infrared wavelength. And that basically lets us create a fingerprint, an infrared fingerprint that we can use to chemically characterize and oftentimes uniquely identify the material underneath the AFM tip. We can also use that tunable infrared laser. We can tune it to a specific fixed wavelength corresponding to a specific molecular bond, for example, associated with a specific chemical species. And then we can raster the AFM tip over the sample to create an image of the distribution of a specific target chemical. So we can then use this technique to answer that previously unanswerable question of what is that. We can take an image with AFM, we can get this unique infrared chemical fingerprint and then determine what the chemical composition is of the material under the AFM tip.
That is amazing. Thank you so much. And you do speak about your work with a lot of passion and love, which is always great to hear. Absolutely. So you use basically an infrared laser and the absorption to measure the cantilever movement. But I guess to get this IR chemical fingerprint, as you call it, you kind of need to combine two very different things. So I would be really intrigued to hear a bit more about how did the idea about combining AFM with IR come about, and what was the early history of really saying, we'll bring those two together to get this chemical fingerprint?
Yeah, the history is really interesting. So as I mentioned, when I was working in the AFM industry, we had this long felt challenge to try to identify the material under the tip, and we tried a variety of different techniques that measured material properties without getting to chemistry. Interestingly, when I was finishing up as a graduate student, I was at a Gordon conference at one time, and I was talking with some mid-career researchers who were talking about, well, what should I do next? I felt like I have mastered a lot of AFM, and it would be interesting to look at other techniques. And they suggested vibrational spectroscopy. And at the time, I just hated the idea because AFM, was used for making these beautiful images, and spectroscopy was used for making kind of ugly squiggles on a paper. It just didn't interest me at all. But that suggestion sort of lodged in the back of my head. And fast forward more than 20 years, after we had tried all of these other techniques for trying to get some insight into what the material was under the tip, we started realizing that spectroscopy could be the potential solution, and started looking for ways that spectroscopy could be combined with AFM. And one of the first people who did some work in this area was a researcher at JPL at the Jet Propulsion Laboratory down at Caltech, Dr. Mark Anderson. And he combined a conventional infrared spectrometer, or what's called a Fourier transform infrared spectrometer, with an AFM, and he demonstrated the basic concept of being able to get an infrared spectrum using AFM tip-based detection, and that got us really excited about the potential. Unfortunately, that particular implementation of using an FTIR was not well suited to getting the best results out of an AFM. The sensitivity was low, and the spatial resolution was not what the market was really looking for. And the real breakthrough occurred a number of years later, led by Alex Dazy, who was working at a university outside of Paris in France. And he combined an AFM with a tunable infrared laser source called a synchrotron, which is a large institutional source, the size of a large building. But he was able to then demonstrate very high spatial resolution and high sensitivity. And that was sort of the proof of concept that you could get nanoscale chemical information with the specificity of infrared spectroscopy. The only problem there was that synchrotrons cost, millions and millions of dollars to build, and they're not portable. So that approach wasn't really transferable to other facilities. So then my colleagues, Roshan Shetty and Kevin Kohler, started working with a laser scientist, laser expert named Konstantin Vodopyanov. And together they put together a benchtop tunable laser that they coupled to an AFM for the first time. And that was what really enabled us to have a vision for a commercial product that would allow that very high spatial resolution and be an instrument that could be implemented in lots and lots of labs all over the world.
It's really interesting to hear where it all started. So you kind of spoke initially about the Fourier transform infrared spectroscopy, or FTIR, and then you also spoke about the synchrotron, which is amazing. And like you say, unfortunately, not everyone has one, as much as we wish that we had. And so obviously AFM-IR solves a lot of these problems, right, that you say. It's probably also a little bit more applicable to samples in terms of it's non-destructive as maybe other systems.
The other key thing that AFM-IR solves is the spatial resolution of conventional infrared spectroscopy. So, one could have asked, well, why was conventional infrared spectroscopy, not solving the problems of being able to map materials with nanoscale spatial resolution? And the reason is that conventional infrared spectroscopy is an optical technique, and the spatial resolution, the best spatial resolution that can be achieved by that technique, is limited by optical diffraction and dependent on the wavelength of the light that's being used. So, for example, if you were to use a microscope using visible wavelengths, you'd be able to get a pretty sharp image with spatial resolution down to a few hundred nanometers. But if you replace that visible light with infrared light and try to do the same measurement, the image becomes blurry because the infrared wavelengths are an order of magnitude longer than the visible wavelengths. So instead of having spatial resolution of a few hundred nanometers, you have spatial resolution of several to many to a few tens of microns. And so, that's very, very coarse compared to the spatial resolution of an AFM. And so the big advantage of AFM-IR was that it was able to provide a measurement of infrared absorption with a spatial resolution not limited by the wavelength of the infrared light, but rather by the size of the AFM tip. Basically enabling spatial resolution to go down to the scale of 10 to 20 nanometers, in some cases smaller than 10 nanometers.
Which is what we all love. We all love good resolution, absolutely. And so now you spoke a lot about AFM-IR, which is absolutely great. But we also know that he obviously developed and absolutely spearheaded optical photothermal infrared technology, which takes all of this a whole step further. And maybe you can explain to our listeners what it is and what the benefit of that technology is.
Yeah, so optical photothermal infrared, or OPTIR, is another analogous photothermal infrared technique that has many of the same advantages of AFM-IR, except instead of using the tip of an AFM to measure the infrared absorption, it's using a typically visible probe beam. So it's using an optical beam instead of an AFM tip to measure the infrared absorption. And so it has pros and cons. The spatial resolution is not as high as AFM-IR because the spatial resolution of the OPTIR technique is limited by optical diffraction. So it's limited to the how tightly you can focus the visible probe beam, which can be down to a few hundred nanometers or so. But the big advantage is it's a much simpler approach. So AFM is an extremely powerful technique, but it's also a very sophisticated technique and requires a certain level of sophistication by the user to get the best results. And the OPTIR technique, while it has somewhat coarser spatial resolution, is more like operating an optical microscope, and so requires a lower level of user sophistication to be able to still achieve very useful chemical spectroscopy and chemical imaging.
Oh, I love this. Yeah, because obviously we could talk resolution all day long, but it's also so important to think about the user. And like you say, if it's like having optical concepts, it's so much easier to kind of move from a normal optical microscope more towards more advanced technology. So that's obviously really important to also keep in mind that users need to understand and be able to use the technology. And so that's really great because it also brings us to applications, because now we spoke about the kind of technology, but obviously there's a whole range of applications. And generally AFM can be used for a wide range of applications, but we want to really focus today on AFM-IR. And so we would love to hear a bit about your top application of AFM-IR.
Yeah, and before moving into that question, I wanted to mention the fact that AFM-IR and OPTIR are really complementary techniques, and in many cases, many labs are using both techniques. They're using AFM-IR when they need very high spatial resolution. They're using OPTIR when they need to image larger areas of the sample, and so it's not uncommon to see publications where both techniques are used. And in terms of applications, there's a lot of overlap in the applications, but the big applications for AFM-IR polymers is absolutely a really big area. Polymers and composite materials, more generally material science, because a lot of modern polymers are blends of different materials. And so AFM-IR is used to map the distribution of the different chemical constituents and specifically characterize them spectroscopically. Lots of applications in the life sciences, looking at proteins and cells, and there's been research into neurodegenerative diseases, looking at things like protein misfolding that are cornerstone concepts related to things like Alzheimer's disease. There's also been a lot of AFM-IR done on semiconductor and 2D materials, and just a huge range of different applications. There are now thousands and thousands of publications using AFM-IR.
Which is always great to see. You also briefly mentioned now that AFM-IR and OPTIR are complementary and can be used as complementary tools or technologies. Could you maybe explain an example where that would be useful and maybe what some challenges could be when you try and do this?
Well, I touched on one of the application areas, and that is related to, for example, different scan sizes. So AFM, of course, is the clear leader for obtaining the highest possible spatial resolution. But when one has to scan an AFM tip over a large area, that becomes prohibitive in terms of time. Where the OPTIR's coarser spatial resolution can let it cover larger areas of the sample faster. And so a common use case in labs where they're using both AFM-IR and OPTIRs, they'll use OPTIR to measure large areas of the sample to understand the sort of micro scale chemical composition, structure, et cetera, and then identify regions of high interest where they want to get higher spatial resolution, and then that's where the AFM-IR comes in.
So that's a great application example. As you mentioned, OPTIR, you scan larger sample areas and then you identify the regions of interest and use AFM-IR to really get in more depth into this with more and higher resolution to get more information. And I think that's also really important because sometimes it's really difficult to know what sample area to look at, and OPTIR can then help you to give you that overview and that global insight into kind of, the context of your small area of interest, which is great. And I guess it's also, especially for new users, really interesting to first get an overview about the material in general and then zone in onto certain regions of interest. So you spoke about, a lot of different application examples, but maybe also for our listeners, is there one kind of experiment or breakthrough that always sticks in your mind as like the main discovery maybe of AFM-IR, that it was only possible because AFM-IR?
Yeah, I think, that's a bit like asking a parent, who's their favorite child. There are so many outstanding papers. But some of the ones I think that have been really exciting are the publications that have the potential to advance understanding related to human health. And so, for example, there have been researchers that have looked at protein aggregation, as I mentioned, associated with the onset of Alzheimer's and Parkinson's and things like that. And there's no other technique that can get the information about the chemical structure, the protein secondary structure with spatial resolution, down to the scale of individual proteins or small protein aggregates. And so because that has the, potential, when you have the ability to look at an onset of a disease at the very, very early stages, that gives us hope that we'll be able to understand the disease mechanisms and understand how to help prevent, stop, or reverse those diseases in the future.
We would never make you pick your favorite child, but obviously technology. So yes, I think it's also, like you mentioned, I think we often forget that these technologies are not just in the lab looking at materials, but actually they have a real world impact, and we need them for like bench to bedside translation. So it's great that you bring that in and say, no, actually we do look, at proteins and for example, diseases like Alzheimer's. So it has a direct implication on human health, which is always great to hear. Thank you for bringing this up. And you also mentioned very briefly before that kind of combining AFM-IR and OPTIR is a possibility, which could be called multimodal imaging, right? But we also love a few other technologies. So maybe you can speak a bit about, what other methods could be combined with AFM or AFM-IR? And do you think that's kind of currently a hot topic, or is it just that we as image analysts love to talk about it a little bit too much?
Well, multimodal imaging is, absolutely a hot topic, not just in AFM, but also in optical microscopy and other forms of microscopy and spectroscopy. And I think there's broad recognition that the more sort of independent channels of information you can use to understand a sample deepens your knowledge and the potential to use that knowledge to advance technology, advance the development of consumer products, advance innovations in human health, et cetera. And so AFM from the very early days has been very strong in different forms of multimodal imaging. So I mentioned previously, the AFM can be used for measuring things like the mechanical properties, like the elasticity or stiffness of a sample, the dissipation through phase imaging, electrical, mechanical, thermal properties, friction, et cetera. And so there are great examples in the literature of people combining the chemical information from AFM-IR with the other multimodal information from the other techniques that I mentioned. And then there's also it's very common for people to combine various other forms of analyses, for example, fluorescence optical microscopy or Raman spectroscopy, and people even combine with X-ray diffraction and in some cases mass spectrometry. And so all of these provide us more information, insight about the samples that we're studying.
Absolutely. And so I'm myself more in like the optical microscopy field. So it's always great to hear, it all has relevance, which is absolutely important. And I think it's also really we can hear clearly that you love your work and you're really excited about microscopy, which is also, great that you're here and speaking with us. But we also love to end our conversations with speaking a bit more about the future. So what excites you about microscopy, maybe current trends, and where would you like to see the future of microscopy?
Yes, I'm absolutely very excited about the future of microscopy and spectroscopy. And one of the things that really excites us is that we look at the publication trends in AFM-IR and OPTIR. We're seeing, very strong growth in the number of publications in those techniques. And so we know that they're being used effectively to enable research in very diverse areas. And so, that's very rewarding. Looking to the future, the big challenges that sort of face the field and provide I think opportunities are to make the techniques more accessible, for example, such that lower levels of expertise are needed to get great results. Things that can make the systems faster, more automated, more economical for certain applications. Those are all areas that are going to enable an even greater impact.
Oh, it's so exciting to hear. Absolutely. And I think especially accessibility of the techniques is so important. If you think about commercialization, the better the software and the manuals and the training, the more people can use it. But you also touched on it that obviously it does need user experience and training to really be able to use these techniques to their full purpose. So it's great to hear that, we still need a human in the whole game. So it was so insightful to listen to you today. And so we would like to thank you for your time. And it's been absolutely great getting your insights. And we hope that you maybe join us in the future again. Thank you so much.
Thank you. It was my pleasure.
So dear listeners, that's all that we have time for in today's conversation. We have covered a wide range of topics, everything from Alzheimer's disease to material sciences. We have focused on AFM-IR, which is a technique that integrates AFM with infrared spectroscopy to deliver nanoscale chemical analysis. And we even touched on a more advanced technology, which is OPTIR, and even spoke about the fact how we can combine AFM-IR with OPT-IR. If you have not already done so, be sure to listen to episode 4, where we talk to Professor Dr. Sergey Kalinin from the University of Tennessee and Chief Scientist in AI for Physical Sciences at the Pacific Northwest National Laboratory. In this 4th episode, he talks about how machine learning in microscopy helps us to automate experiments, the data we need for successful AI, and how we can get the most out of data that we have already acquired. So thank you so much for listening. Until next time, goodbye.
This podcast was sponsored by Bruker Nanosurface and Metrology Division. Bruker is a manufacturer of scientific instruments for life sciences and materials research, as well as for industrial and applied analysis. If you'd like to learn more, please visit www.bruker.com. This podcast was produced by Keep It Science.